Really warm welcome to the Department of Public Health Friday seminar, and we're delighted to have Siouxsie Wiles with us today, and she's got the great title, The End of Modern Medicine, and I think it's part of that strand in public health of sort of mapping out potential dystopian futures and what we can do to avoid getting there, hopefully. And Siouxsie, of course, in many ways needs no introduction because she's really, as well as being one of our foremost public health researchers or antimicrobial resistance researchers, she's also one of our foremost science communicators. And I'm sure you've seen her or heard her on national radio. And she was this year one of 3 finalists for the Kiwibank New Zealander of the Year award. And I thought it was wonderful to see a top scientist in the running for that award, and it was very well deserved that being on the shortlist there. So over to Siouxsie.
Thank you. Kia ora, and thank you very much for coming this lunchtime. So let's talk about the end of modern medicine. Really exciting. I just want to start though with an acknowledgement to the people both in my lab now and in the past, and also our kind of collaborators in various places. Most of our science I'm not actually going to talk about. I will talk about one of our projects at the end. I was more sort of going to give a kind of introductory talk around antimicrobial resistance, but obviously all of the stuff that I do would not be possible without this very large list of names, so I just want to acknowledge them. All right, so my talk is kind of divided into 2 parts. The first, we're going to talk about the apocalypse, and then the second part about what we're trying to do to stop the apocalypse from happening. Obviously, we're just a small part. It's probably going to happen anyway, but just show you what we do to keep ourselves going. And then I've hopefully have left plenty of time for us to have some discussion at the end. Although being an academic, you know what that's like. Hence the timer. All right, so the apocalypse. So I'm originally from the UK, but I met my Kiwi husband while he was studying in the UK. And then when we had a child, he did that thing that happens to Kiwis where some gene gets expressed and then they want to move home and have their child grow up with sand between their toes and things. And the way he kind of sold it to me was that, you know, and obviously we'd been together quite a long time before our daughter was born. I'd been on holiday to New Zealand many times. And so he sold this kind of, it's going to be an amazing lifestyle. It's like Auckland, the weather's great, apart from the humidity. And you know, we're going to be in this beautiful city of sails, or this beautiful water. It's going to be great. And I was like, actually, he didn't need to say any of that. All he needed to say was glowworms, and I was like, there. But anyway, he didn't mention the glowworms. But that's actually secretly the reason that I came. We can talk more about that later. So he sold me this vision of New Zealand, and like I was there. I'm like, yep, let's move our family, and we did. And I guess being a fairly wealthy white middle-class person, we kind of have this fantastic lifestyle. So we live in Ponsonby. This is one of our kind of fairly local beaches. In December, I decided to start getting out on my bicycle more. So we sort of did a lot of cycling around just to all our local beaches. This is a sunset at one of those beaches. And it's just like, yeah, we're living the life. This is kind of amazing. But obviously when I got here, I guess it took a little while to settle into life into New Zealand and then to kind of find out what life was really like. And actually it was kind of thanks to researchers here and Michael Baker that I really got a feel for what life was like for some New Zealanders in this sort of area that I'm really interested in, of infectious diseases. And for me, this was a really pivotal paper, the work that was published in The Lancet about our infectious diseases burden, that really changed my thinking about my research, actually, my relationship with my research. And so for those of you, I mean, you must all be familiar with this paper, that basically they're looking at the data for hospitalizations and showing that the hospitalizations from non-communicable diseases went up over the 20 years that they studied the data. And I think it was by 7% or something, and then the data for hospitalizations for infectious diseases also went up, but by like a huge amount more. It's just kind of, we live in this insane place where it's this beautiful country that has this growing burden of infectious diseases, which is not what countries like us should be like, right? We have this sort of idea that as countries become more developed, their infectious diseases rates go down, more people get non-communicable diseases. And for New Zealand, that's not really true. And that's been a really striking thing for me. So the sort of looking at all the numbers, it's like the equivalent of 1 in 4 people who are hospitalized at least overnight in New Zealand are there because of an infectious disease, which just seems wrong, really. So a year or so ago, I just had a play with some of the hospitalization data for the year 2000 that ended in 2014, looking at, and if we look at infectious diseases, what are the reasons that people are there? And I divided it into those that were suffering from a bacterial infection, those that were suffering a viral infection, and then fungi and other agents. And of course, this is just the people who have this coded on their hospitalization data. There's actually many more who will be battling an infectious disease that is not even kind of on that data. But you can see in New Zealand, the vast majority of things that are there are bacterial infections, which is good because I'm a person who likes bacterial infections. And when we then look at that data and break it down a bit further, I've put in TB and the mycobacteria because there's something that I'm kind of interested in. So we get a little small numbers of hospitalizations caused by those, a very small number by sexually transmitted infections. They're really sad, although small little thing from vaccine-preventable infections. But you can see that the vast majority of those hospitalizations are due to these sort of 2 categories, food and waterborne diseases and strep and staph infections. And so all the data that's sort of pointing now to the fact that we have higher rates of many of these diseases than countries that we would like to benchmark ourselves against. And that's, again, like an appalling thing. And what's really striking to me for those 2 categories is that this is all about how we treat people and how we treat the land. Those are the kinds of diseases that pop up when we do those 2 things badly. And I think we can really categorically say we do those things badly here. Striking, right. So that was sort of hospitalizations, but obviously there was a huge burden of infectious diseases that would never appear in that data because people are not hospitalized. And so I've put condoms up there because sexually transmitted infections are actually one of those where we have a massive burden that is kind of hidden to everyone. So the data seems to suggest that we have got a huge numbers of people who may well be infected with chlamydia, which for most people it's asymptomatic, so they wouldn't even know it. But the long-term consequences of an untreated chlamydia infection are infertility. And so for me, this seems to strike as we have some diseases that we don't even know that we have, and they are going to potentially have a massive impact on our future ability to reproduce. And, you know, we all need babies and young people essentially to pay everybody else's superannuation, right? So this is a really big issue, and it's one that we're not thinking about, or I don't think young people are thinking about at all. And it's kind of ironic, right, that you spend your teens and your 20s trying not to get pregnant, and then you try your 30s and 40s to get pregnant, and we're kind of going to make that even harder. And so I think we really need to be having conversations around why don't Kiwis want to use condoms, you know, sort of these sorts of things. Okay, so yeah, we're living in this kind of amazing country that also has these incredible rates of infectious diseases, many of which are even hidden. And the real problem with this is that we're running out of ways to treat these infections. So this is a real kind of scare. And what I want to do is just show you this really amazing video that was filmed by a scientist at Harvard called Michael Baum. And he's just done this really cool experiment that visualizes this problem that we're having with the fact that our medicines aren't working anymore in a really, really beautiful way. So let me play. So what Michael has developed is what he calls the mega plate. So this is a vast petri dish you grow bacteria in. It is like 1 meter long by ½ meter wide. And it's divided into sections. And so in each of those sections, he puts the agar, the jelly that the bacteria will grow on, but he puts different amounts of an antibiotic. So on the very end, he puts no antibiotic. Then in the next section in, he puts just an amount that would normally kill that bacteria. Then he puts 10 times that amount, 100 times that amount, and then in the middle, 1,000 times that amount. Then what he does is over the top of that, he puts another layer of this agar, but this one's a bit sloppy. So if a bacteria has a tail, they can swim through it. And then he essentially adds bacteria to the ends where there's no antibiotic. He sets off his time-lapse camera and then he watches them grow. And so you can see they're growing, that's the white, grow really, really fast until they get to that end where there's antibiotics and then they stop. And then the only ones that can survive there are essentially mutants that can survive in the presence of the antibiotic. And so you see these mutants appear, they start to grow out, they sort of start to compete with each other, it's quite cool. And then usually what happens when they get to the next barrier is they'll stop again until there's one that has the mutations that allow them to survive in that amount of antibiotic, and then they will start to grow and compete. And this happens step by step until finally we get into the middle of the petri dish, 1,000 times the concentration of the antibiotic that would have normally killed them, and they can grow in the presence of that. So this is kind of evolution in action, quite exciting being able to visualize that. And then, of course, they've done really cool science where they basically can plot what the mutations are that allow the bacteria to get to that stage. So I really, really like showing this when I give talks, primarily not to you as an audience, but to usually to members of the public, because there is a real misunderstanding about what antimicrobial resistance is. Lots of people say to me, oh, I don't take antibiotics because I want them to work when I need them, sort of implying that it's us that become resistant to the antibiotics and not the organisms themselves. So I think this is a really fantastic example of showing, look, it's the bacteria. They mutate, they become resistant. In this case, it's E. coli, and that happened in 11 days. So in 11 days, that bacterium can become resistant to 1,000 times the amount of antibiotic that would have normally killed them. And it's kind of mind-boggling for a lot of people. So what it also shows, of course, is that this is those are mutants that arise essentially by chance, right? So we're all in this room mutants, you know, mistakes are made when our genes are duplicated, our genetic material is duplicated, and the bacteria are no different to this. And of course, we all also have really good processes in place to kind of try and stop most of those mutations, because we don't want them to, you know, like we obviously don't want to just end up as a mutant goo that can't function. But mutations that happen are, you know, having some get through is a good thing, because that's how we get variation. It's how we get evolution. So the point is, of course, that sometimes those mutations, just by sheer chance in the bacterium, will allow them to become resistant or be resistant to antibiotics. And it's really this kind of random process that is an inevitable just fact of life that has kind of got us in the pickle that we're in at the moment. So these mutations happen by chance, but it's really, we've been, I guess, living in a world over the last sort of 50 or 60 years that has been providing an environment for those mutants to thrive. And that's been our use of antibiotics and I use our as like humans rather than New Zealanders, but everybody around the world who's been using antibiotics both in human medicine and animal medicine, in agriculture, horticulture, or aquaculture, wherever we've been using these things as humans, we have been essentially putting these antibiotics into the environment and then allowing those organisms that are resistant, essentially by chance, an opportunity to thrive. And so this just is a little kind of summary of a place that shows all of the places where antibiotics are used and where resistance can emerge. So we've got, you know, a community with hospitals and houses and people. We've got agriculture and horticulture. We've got aquaculture. We've got, I guess, factories where antibiotics are made. And what this all means is that just like tons and tons and tons of antibiotics and their kind of building blocks have been present in the environment, I guess, in increasing numbers over the last probably 50 or 60 years. And all these bacteria are basically like, excellent, we shall thrive and conquer, which is basically the state that we're in at the moment. So bacteria can become resistant by chance, but they also have a few other little tricks up their sleeves which allows them to transfer that resistance to other organisms. So essentially they can do one of these three things. So transformation is the process of taking up genetic material from their environment. So say, for example, there is an organism that is resistant that has died and released its genetic material, then other bacteria can take that up and say, excellent, thank you, we shall use that. Or they can get infected with viruses, and viruses can essentially move, almost by mistake, can move bits of genetic material around, so they can move a resistance gene essentially from one organism to another. And then they can have the equivalent of sex, conjugation, where they build a bridge between each other and essentially exchange material that way. So we have this process happening all around us, completely natural process, things are becoming resistant by chance, and then basically sharing that thing around. And this has happened forever, because most of the antibiotics that we use in medicine are compounds that are naturally derived from microorganisms. They're essentially used by microorganisms to attack each other as they fight for space and nutrients. So they've been doing this a long time, but we've essentially been pushing them into the environment and kind of allowing these sort of superbugs to thrive. And so this has been happening since, in sort of increasing numbers since we started using antibiotics in clinical medicine in like the 40s. But for a long time it didn't matter, because we had this age where after the discovery of penicillin and other antibiotics, where we had this sort of like gold rush of looking for these incredible compounds. So this is essentially a timeline where each of those little triangle arrow things is the discovery of a new class of antibiotics. And so you can see that they're sort of centered around the sort of 50s, 60s, and 70s. That's when all these amazing new classes were put into the clinic. And so resistance has been happening all that time, but when a bacteria became untreatable, it was okay, because they just basically, or when it became resistant to that particular antibiotic, the doctors would just go back to the cupboard, right, and pick up another one and go, hey, we'll use this one now. So that's okay, so great. So we've been basically taking stuff out of the cupboard over the last so many years. The problem is that we haven't been refilling the cupboard at the same rate. So this little area here called the discovery void is essentially when the pharmaceutical companies pulled out of antibiotic discovery and development, focusing their attention instead on, cures, treatments for more non-communicable diseases. And so, but of course, resistance has still been happening. And so now we've reached the stage where essentially the cupboard is empty. There is nothing being put in it. It's unclear whether there's anything going to be put in it, or if there is, how long it's going to take to start refilling that cupboard. But we now have a whole bunch of organisms that are actually, if not untreatable now, almost untreatable. And that's quite scary. It's so scary that the World Health Organization finally got their arse in gear, and in 2014 released their first kind of survey of resistance around the world, essentially concluding that resistance is everywhere, but actually most countries don't even have the means in place to even survey it. Most countries don't even know what they have. And so Margaret Chan, who was Director General of the World Health Organization at that time, she called antimicrobial resistance the end of modern medicine as we know it. Because of course antibiotics are not just used to treat infectious diseases, they're used to prevent infectious diseases in vulnerable patients. So all of those who are having surgery, who are having chemotherapy, all of these people, they're used to treat that. And so it's kind of amazing thinking what life might be like when we don't have those treatments. I guess that we will still do them, but people will probably start dying in large numbers from infectious diseases. Okay, so what does this mean? Well, there are basically a few organisms now that are kind of on this utterly terrifying, untreatable sort of category. They're fairly rare still in countries like ours, but we are essentially at the beginning of an epidemic. So this is a headline from USA Today. It was the first case of a completely untreatable superbug in the USA. The woman had broken her hip while in India, had had some treatment in a hospital there and then moved back to the USA, or gone back to the hospital in the USA. She brought with her a rather nasty organism that was resistant to the 26 antibiotics that the doctors in the USA tried to treat her with, and there was nothing left and she died. They're looking at the bacteria. There may well have been one drug that it was susceptible to, but it's a drug that isn't licensed for use intravenously. So it's only allowed to be used orally, I think, for cystitis, and they basically couldn't give enough of it to her for her to survive. And it's one of these carbapenem-resistant Enterobacteriaceae, so a class of bacteria that can often be carried by people with no symptoms at all, but have essentially picked up a whole bunch of resistance genes that are making them very difficult to treat. The cases that we see here in New Zealand and in most places are still treatable, but they're treatable with what we call a last resort antibiotic. So it's one that's a last resort because it was left in the cupboard because it was pretty toxic to people. So everyone was like, okay, we won't use that one, it's not particularly nice, it got left in the cupboard. But unfortunately, agriculturists found it in the cupboard and started using it in pigs. I think it was mostly pigs in Europe and in China. So resistance has emerged and is now spread to the strains that cause disease in humans. And so, we don't. So this is kind of an emerging thing that basically the cases we're going to start to see now will be resistant to that too. And then we're going to end up with more of these kinds of things. So we have seen this in New Zealand, not the completely untreatable ones, but just in February this year, our National Burn Centre at Middlemore Hospital had what they call an outbreak. There were only like 3 patients. I'm not sure that really constitutes an outbreak, but anyway, but it's really serious. I mean, these are really vulnerable patients. And so when, if their burn gets infected with one of these organisms, it's very, very difficult to treat. I mean, it must have been really quite scary. What actually happened, so this is the data that ESR put out on the web. I love ESR and their stuff. If you know where to look, there's all sorts of exciting things. So this just has it by patient number, the species of bacteria that they isolated, the resistance gene that they isolate that's from those organisms, the date of the specimen. It's not clear from that data whether some of them were just swabs or whether they were actually ones from people who were infected, and then the likely place of acquisition. So the feeling in New Zealand around these strains is that they're not here, they come in from overseas, and so usually from someone who's been hospitalized overseas. So that first person, patient 35, I guess was swabbed maybe on entry into hospital and was found to have 2 organisms. They had previously been in hospital in Tahiti, so they reckon that's where they got it. So you can see they had a Klebsiella and a Pseudomonad, and they had both had this NDM1 gene. Patient 37 then was essentially had no history of travel, but was in the same ward as that person. And you can see what they ended up finding over a course of a few weeks was 5 different organisms that have the same resistance gene. And so what I've highlighted is that they have one organism in common. They have Klebsiella pneumoniae. So what looks like is likely has happened is that the Klebsiella strain has moved from patient 35 to patient 37 somehow. But then in patient 37, that organism, Klebsiella, has moved its resistance gene to the other 4 species in that person. And so this is a really good example of showing that how they can then spread that resistance gene around. And what you now have is a patient with 5 different organisms which will have completely different profiles of which antibiotics they're resistant and sensitive to, because they're all really different organisms. And if they get into the burns of that patient, it will be very, very difficult to treat them. Both patients were treated and have been discharged. And then there's a really interesting little one down the bottom, 39, that on the table in ESR they've called part of the same outbreak, but they have a different resistance gene. And they also didn't seem to have had any travel overseas. They just put Middlemore Hospital ? As a place of acquisition. And so this is making me wonder whether actually we probably have these strains here, but they're just being carried by healthy people, and that's quite scary. But I think that's quite good evidence for that. So this just shows you the number of these organisms essentially since 2009. So we used to get sort of like 1 or 2 cases, but we're now starting to see more like 50 or 60. And so this really is just sort of the beginning of this epidemic. And it'll be really interesting to see how this plays out. Interesting as in terrifying to see how this plays out. Okay, the other big one is Staph aureus and methicillin-resistant Staph aureus. So this is this bug that we have huge rates of here, especially causing horrible skin infections in our young children. It's been really interesting to see how in New Zealand's response to this infection, which is to treat children with an antibiotic cream, has basically selected for a much more resistant strain to be circulating in New Zealand. And so this just shows, all this shows is by year, the different varieties of the Staph aureus that have been predominant. And all I want you to look at, so the colors show the different kind of ones, and you can see how the dark blue one is increasing in number until now it's actually the predominant strain here. And that's the one that is resistant to the antibiotic in the cream that is used to treat staph infections. So our use of cream to treat staph infections, because we have hideous rates of the stuff, presumably because kids are living in horrible housing, is basically driving now this resistant strain to be the one that's predominant. And I think we're actually even exporting this one overseas now. We're sending our resistant bug to the rest of the world. Yay, New Zealand. Okay, and then this is one we've had a few little cases of, but it's as one to watch. So super resistant gonorrhea. There's been plenty of cases in Japan, in Australia, in the UK. And it's another really interesting one, because the case in the UK that happened really recently, so people who have gonorrhea, like this, so this dude basically went, he was in Japan, he did some things he probably shouldn't have done in Japan, and then he went home, and then he had sore genitals, so he went to the doctor and got swabbed and given some drugs. Then they found out that they swabbed his throat and his genitals. They found out that he had this gonorrhea in his throat and his genitals, and then and it turned out to be a really resistant one. They had to give him quite a lot of treatment. But obviously the way they do it is people have to come back and have their swab to see whether they're clear. So he came back, was swabbed again. I guess that takes some time. So he obviously went home, didn't stay at the hospital or the sexual health clinic, and then was asked to come back, because while his genitals were clear, his throat wasn't. And apparently it took several months before he came back for more treatment. And so this kind of really interesting idea of, you know, those who are carrying these organisms and potentially spreading them but have no symptoms is quite frightening. So who knows who he might have spread his super resistant gonorrhea to, just kind of having fun without using protection. So if you're interested in this area, I don't know if maybe I'm preaching to the converted. I spent my summer last year writing a book about it, because I wanted more people to know about this. And so, yeah, we called it The End of Modern Medicine with a question mark, mainly because we just didn't want to freak everybody out. But yeah, it's a kind of a short book, and it was a really interesting and kind of a really good experience, I think, to sort of put all of that stuff into one place. Okay, so how do we prevent this crisis? Clearly, we need to use the antibiotics we've got more wisely. So this is a big push, especially in hospitals, I guess, but it needs to be done in the community too, around the sort of antibiotic stewardship. We clearly need to be using vaccines and other preventative measures, so stopping people from getting infections. Of course, the problem with most of the diseases that are the problem is that there aren't any vaccines. We really need to be tackling our infectious diseases rates. And it's unfortunate that this is something that seems to not be in the work plan for how we're going to tackle antimicrobial resistance here in New Zealand, and that's a really big problem. And then obviously we'd like some new antibiotics and vaccines, again, something that's not in the work plan here in New Zealand, because we're considered too small a country to be kind of, I guess, to use our resources on that. So in the last few minutes, I just want to talk about what we're doing for this, because I'm essentially a basic scientist with an interest in microbes and killing them. And so this has sort of been, how can we help in this search? And what we're doing is working with Manaki Whenua, who are one of our Crown Research Institutes, who have this amazing collection of fungi. So they have had people who have been collecting fungi from around New Zealand and the South Pacific for like 50 years, and they're all basically stored in liquid nitrogen in Auckland. And a very chance meeting with one of the mycologists there was like, we've got all these things and they've never been screened for antibiotics. The background being that penicillin comes from a fungus. So the idea is, given that New Zealand has been geographically isolated for so long, and you know, we have kiwi and all sorts of cool things that have emerged, why would our fungi be any different? Do we have some cool fungi with novel chemistry that might be producing antibiotics that haven't been discovered before? So we're going to find out. So basically the collaboration is with Bevan, who is the essentially the custodian of this collection at the moment, curator, in my lab, where we basically make Bacterial Glow-in-the-Dark. I'll talk a bit more about that in a second. And then Brent Kopp, who's a chemistry professor at Auckland, whose job is basically, if we find anything that kills, to try and figure out what it is that's doing the killing, and the hope is that it's not just penicillin over and over and over again. It is a bit needle in a haystack, but you've got to try somewhere. So this is where I come in. Essentially, I've made my career making bacteria glow in the dark, and this is kind of cool for various reasons. It's kind of cool. The reason that applies to why we can apply it to cool stuff is because they'll only glow when they're alive. So it gives us a really quick measure of whether something is dead or alive. So essentially, what the bacteria are doing is producing a chemical reaction. So they essentially, we have this gene operon called the Lux operon. If we put in that into bacteria, they'll basically make this enzyme that converts an aldehyde to a fatty acid with the production of light as one of their byproducts, and then they can basically recycle that fatty acid back into aldehyde. to make more substrate. It requires FMNH2, which basically means the bacteria need to be alive, and it requires oxygen. And so my career has just been like anything that stands still long enough, can we try and shove these genes into it? And it's kind of cool, we can. So this just shows you essentially the more bacteria there are, the brighter the light will be. So we can basically get a feel for how many numbers there are just by putting the bacteria in a machine and pressing a button rather than plating them out and counting bacteria. So I guess it's the lazy microbiologist way of doing the microbiology. And then the other one is just showing you this, are they dead or alive? So basically taking a 96-well plate, adding little amounts of bacteria, adding little bit amounts of chemicals, and then saying, are they dead or alive? So it's kind of a cool, non-invasive way, again, of kind of doing those sort of experiments. My timer's going to go off in a minute. I'm going to give myself more time. It's naughty, isn't it? Okay, I think we're all right, though. Right, okay. So what we essentially do is we grow the fungi on petri dishes. We punch little holes in those, in where the fungi are growing, and then we add our bacteria that glow in the dark to those petri dishes. And so here what we have are each of those little circles is the bacteria, but we've got 4 different species of bacteria growing in there. And then we wait a few days. We put them in our machine to measure the light every few days, and then we're looking to see, does the light stay or does the light go? Is the fungi producing something that can kill the bacteria, and is it producing something that kills all the bacteria, or is it producing something that's killing specific species of bacteria? Because ideally what we want are some narrow spectrum agents that aren't just like kill everything, but that sort of will kill things individually. So we're testing against E. coli, Klebsiella, Staph aureus. We're also testing against a couple of fungi now, all sorts of things. So we sort of have this little like machine going now that's sort of testing all these things. We also have some really quite resistant strains of some of these bugs that don't glow in the dark yet. So we also do these zone of inhibition assays where we basically grow the bacteria on the petri dish, add the fungi plates, and then we look for zones of inhibition where the fungi are stopping the bacteria from growing. So again, in this way we can say, does it kill staph, does it kill resistant staph, or does it kill sensitive staph, that kind of thing. And then we've got another kind of cool little assay where, so once we've found something that kills, we then freeze dry it to make an extract out of it, and then we can run them on these chromatography plates, so to try and separate those proteins out, or whatever's inside of it, and then we dip it inside of the glowing bacteria, a little pot of bacteria, and then we can look for light, and then we can see, is there something there that's doing the activity. Unfortunately, the machine that you need to then take that spot off and identify it is very, very expensive, so we have to use other methods. More laborious and cheaper methods than that. Yeah, so we've screened probably 700 or so at the moment. We've found lots that have activity. We've found lots that have activity that was already known about, but maybe not known to be produced by fungi. I saw the chemists on Tuesday, and there's something exciting, but they won't tell me any more than that. So watch the space. But what I have to do is acknowledge how this project started. So this project happened thanks to Kim Hill, Lego, my hair dye, and then this lovely man in the corner, Peter Buchanan. So Peter Buchanan works for Manaki Whenua. He is a fungi nut, and he was listening to me being interviewed by Kim Hill several years ago. And we talked about my hair, we talked about bioluminescence, we talked about my love of glowworms and fireflies, and Peter was listening at home, and he was like, oh, this is really interesting. I should get in touch with Siouxsie because we have fungi that glow in the dark and she'll probably want to know this. He was right, I did. But he didn't get in touch with me. What then happened was a little while later, so I'm a big Lego fan, like honestly, actually fan is not the right word. I'm obsessed with Lego. I spend all my money, I don't buy clothes and shoes, you can probably tell I spend it all on Lego. Anyway, so I was at a Lego kind of exhibition thing, and he was there with his son who also liked Lego. And so he spotted me thanks to my pink hair and came up and said, oh, I need to tell you about this glowing fungi. And we kind of ended up having coffee and chatting. And then he told me about more than just the glowing fungi. He talked about the collection. And that then led to the collaboration with Bevan. So I have to thank him, Leo, and my pink hair for getting this project off the ground completely. So it has no government funding. We have tried for many years, but it's basically, I think, a kind of fishing expedition. So we essentially get the public to help fund us. So we ask anyone who is looking for an unusual birthday or Christmas gift to consider, rather than a pair of socks, to sponsor one of our fungi. We are not fussy, like $20 buys us a set of petri dishes. 250 is how much it costs to take each of our fungi through the screening. So yeah, we get people kind of signing up. This is a website that we've got up online on the university's webpage for people to sponsor. And then I do some stuff with artists where we have a bacteria that glows in the dark that isn't harmful. It's naturally glowing. They make, obviously, because that would be bad to expose people to nasty things. So they make works of art by basically drawing on petri dishes with this bacteria. We take photographs of the works of art and then we put them up on the site called Redbubble, where you can turn them into t-shirts and stuff. And so some of the proceeds of that, it's probably only been about $200 over the last 2 years, but you know, that's like 10 stacks of petri dishes. So as I say, we're not fussy. Yeah, so that's basically this project. Watch this space, and I'm very happy to answer any questions. Thank you very much. At least a few minutes for questions.
So we have time for questions, and if you could just introduce yourself and speak into the mic so that we've got quite a few people who are joining us from a distance, and then they can hear your questions. So, Nick Wilson.
Yeah, Nick Wilson, University of Otago here. Great talk, thanks a lot, and it's great you're exploring, looking for new antibiotics. But in terms of protecting the ones we've got, have you got any comments about the use of antibiotics in agriculture? I mean, including, I mean, it's not just livestock agriculture, but I mean, tetracyclines are sprayed on fruit trees and things.
Yeah, well, so yes, I have lots of thoughts. I've got to be very careful about how I vocalize them, because I have had an email recently from the poultry organisation saying, you have to come and see us and we'll tell you that we don't use antibiotics. It's quite possibly a definition thing. There's some things that, yeah, kind of they are antibiotics, but they're not called antibiotics. Anyway, what I was amazed, so during the zespri, the kiwi fruit canker outbreak, so that's kiwi fruit disease is caused by a bacterium very closely related to Pseudomonas aeruginosa, which causes problems for cystic fibrosis patients, burns patients. And so zespri went around the country talking to scientists to find out, you know, how could they, you know, how could they solve this thing? And I got really excited because I was like, actually, if you don't, they didn't really even at the time know how the infections happened. I was like, we could totally make that by glow in the dark. We could get some kiwi fruit vines. We could image where they're going, how they enter the plant. And they were like, no, we're not interested in that. So I was like, okay. And what they said was, you know, what you're kind of proposing is too long term. It's kind of, you know, we want something now. And we know we can just use antibiotics. And my colleague and I were like, you're going to do what? And they said, yeah, you know, we can just spray this stuff. And we were like, well, you need to be really careful. Do you know what antibiotics you're using? Because what we really don't want is you spraying all the kiwi fruit vines with antibiotics that are used in human medicine. They were like, yeah, fine. And then, of course, I didn't hear anything else. And then when I was preparing the book, I read all of the documentation from MPI around what antibiotics are used in agriculture in New Zealand. And we actually don't know. We have data. The only data that's collected is sales data. So we know what is sold. And then by that, you can kind of work out what it might be used for. So they don't collect any of that stuff. But what was really clear was around the PSA outbreak, there was a massive spike in the number of antibiotics that are used in horticulture that were purchased, one of which is specifically used in horticulture, not human medicine, but the other one is one that doctors use. And it was just like, wow, they did it anyway. And I guess this is why this idea of One Health is really important, that people who are working in human medicine and people who are working in crops and things talk to each other about what they're doing, because we really shouldn't be using the same kinds of antibiotics in both things, and not just antibiotics, antifungals, all of these kinds of things. So yes, I have feelings, but and comments, more just kind of horrified that we're not talking to each other more about this. And the fact that they went ahead and did it anyway. It's, yes.
Thanks very much. Really, you know, fascinating stuff. So, and I can understand your love of bugs and mushrooms and, you know, glowworms. But especially that, following on from the last conversation with Nick, sorry, I'm Louise Delaney, also Department of Public Health here at the University. I'm interested in law, and I'm also interested in information. And it seems that we don't, from what you've just said, have a legislative mandate for collecting information, which seems a basic prerequisite for addressing any issue whatsoever, yes, and especially one which is going to mean the end of modern medicine. So, yeah, so I'm just really asking for your thoughts on legislative frameworks for information. Secondly, legislative frameworks for looking at this in a sort of a comprehensive intersectoral framework, which seems necessary in New Zealand. And most importantly, a global framework. What are your concepts and sort of thoughts on all of those?
Oh my God. All I'll say, so I think antimicrobial resistance it's like climate change, right? I mean, it's a global problem. We can all try and do what we need to do locally to resolve things, but frankly, you know, you just need to get on a plane with these bugs in your gut or up your nose and you've moved them somewhere else. So we need to be thinking about this globally. And I guess the issue is that while there are some really high level stuff going on, it's really unclear to me how much that's percolating down to actual action. So there are clearly countries like, so I think it was about 2016 maybe that there was the Global Assembly that said, right, okay, this is a problem and everybody who signs up to this needs to have a plan in place. And they set the deadline as sometime like August or something, or May 2017, I think, is the deadline for when those countries that signed up had to have their plan. And so I was kind of eagerly awaiting this. The European Union have been doing quite a bit of stuff. Obama was trying to do a lot of stuff around how can we incentivize companies to come back into this sort of discovery space. He must have been, I'm not sure whether he was doing some of the more that kind of stuff. And then in New Zealand, actually what we got was a kind of plan to make a plan. It was really interesting that there was a real clear, in New Zealand, we should be doing stuff in this one health approach, we should be sort of all together. And then what's happened after that plan to make a plan is they're now doing the plan thing, but they've split again. And so we've been split into a health group and an animal group. And there is a representative on each one. So there's someone from MPI who sits on the health one and someone from the Ministry of Health on the other one. But it's just, oh God, maybe I'm not actually allowed to talk about this because I'm on it. But it just seems like a bureaucratic nightmare that nothing's actually going to get done. Like you just need to bang everyone's heads together and say, what do we need to sort out? Because we were asking the MPI guy about this. So, you know, what can be done about regulating use in animals and stuff? And there was just sort of a because he's like, the rules are different, the legislation is different around that sort of thing, and it just seemed mind-boggling. But I'm not sure how quickly something's going to be done. And this is, I think, the thing we need to impart the urgency of this. But you know, I feel like we are where climate change was maybe 10, 15 years ago, where people are like, yeah, this is going to happen, but it's not happening on a grand scale yet. And maybe here in New Zealand, it's not really going to strike people as being a really bad thing until it becomes too dangerous for the All Blacks to play rugby, right? Because they can't have surgery for their broken bones, or if they scratch their knee, they might end up amputating their legs or something, you know. It kind of feels like it needs more urgency. So I totally agree with you that the law stuff would be useful, but I'm not even sure who's actually looking at that. I don't even know whether within these groups they're thinking about it. And that is a worry.
There have been some proposals for international legal framework.
But how that then filters down, yeah.
George Thompson. Just taking from your last two answers and trying to put words in your mouth.
You can try.
What I take it is that perhaps a agency that would have control of the use of antibiotics and the release and usage and so on, so that they would actually have powers and teeth to say to MPI or other agencies, yes, you can, or
no, you can't. But of course, it's more complicated than that, right? Things are always more complicated than that because people need the antibiotics, you know, they need access to the antibiotics they need. I guess there's a huge amount that's not known about what actually is the best treatment for things. You know, we probably use a whole bunch of antibiotics in ways that is less than optimal because the science isn't there about, like, because the studies have just never really been done about how you would use them more optimally. And one of the really big issues as well is around, certainly in human health, around diagnosis. Like, how do you really quickly diagnose whether someone has a bacterial infection or a viral infection and whether they need antibiotics or not, and which antibiotic they need? And it's really interesting that in the UK, that's become the focus of one of their really big funding challenges. So it's called the Longitude Prize, and it's kind of a hark back to the 300 and odd years ago when they set the prize to find longitude so they could get an advantage at sea, right? They've done another one on the kind of anniversary of that. They put it out to public vote about what the topic should be, and antimicrobial resistance was the one that won. And the project is it's something like £10 million is the prize pot for the first group, and it's open to anybody all over the world, academics, companies, random people in their garage. If you can solve the problem of how to really quickly do point-of-care diagnostics, how to differentiate the stuff so that you can actually give people what they need when they need it, rather than relying on them possibly coming back 2 months after they've been spreading their resistant gonorrhea around. I mean, so there's like this whole, sorry, this is a bit rambly, but it's so complicated, right? So complicated about how you deliver what's needed, whether you're delivering the right thing, because the science might not have been even done about what the right thing is. Yes, we should be restricting it, but I don't even think we know what to restrict, and then of course we have the other problem of people who need the antibiotics who don't get them because they don't have the right access to healthcare. So it's a big mess, but most complicated problems aren't they, right? And I guess what I'm hoping is, by writing the book, was, can we just start talking about this some more? Because we also need to totally be talking about how do we reduce our rates, and that seems to be missing from the antimicrobial resistance thing. Like, nobody's really talking about actually the whole public health stuff that needs to be done to make sure that we're reducing these things. Because yes, we can find another drug, or yes, we can develop a vaccine, but if you haven't removed the underlying drivers why we have these things in the first place, something else is going to pop up and take its place.
One more.
Hi, that was really interesting, thank you. I just wondered, people have been talking about this since at least the 90s, and quite a lot, and you're kind of talking about it as if it's all just a mess and it's all just kind of happening and diagnosis is hard and so on. But we know that, doctors have colluded with big pharma, they've colluded with tobacco companies and so on. My understanding was that doctors prescribing of antibiotics wasn't really going down in the way that, and this is in the UK, that it was hoped it would when they understood that over-prescribing was a problem. So I just wondered, isn't it time to think about regulating doctors, and isn't there a bit more we can do than what you're suggesting? And you're kind of like your universe seems to be full of confused players rather than players who just, except for perhaps for Zespri, who just don't give a toss.
Oh, okay. I think it's more complicated than that. So there is real research around patient demand and those who are coming in, and it's human patients, but also humans who are taking their pets to the vet, right? This expectation that I want something, I want a script with something. And there are definitely doctors who cave to that, who are like, oh my God, I just need this patient out of my clinic. I will give them something. So I think there are degrees of all of that. I think there are absolutely degrees of the kind of, well, we know we're causing a problem, but we've got 10 minutes, we need to get this person out. Right down the spectrum to those who actually don't know, who are too busy to know. I mean, it must be very hard. I kind of feel for GPs, right? They have to kind of keep on top of everything. How do you kind of do that? So I do think it's complicated, and I think it runs the whole thing, but I don't think everybody who's doing a bad job is doing a bad job deliberately. I think there are those who are doing it deliberately, but not everybody. Am I mistaking? I mean, you're right. We have known about this thing for a long time, but I mean, because I remember being taught about MRSA as an undergraduate in the 90s and thinking, gosh, what's going on? And then you're like, oh, nothing really bad has happened yet, right? Because it's just been bubbling along. That's why I say we're kind of like, we're not really fully feeling the effects of it yet. We haven't been feeling the effects of it because there was always something else left to use. But I think it's becoming more urgent. And the really scary thing is that even though we've known about it since the 90s or 80s, in fact, we've known about it since Alexander Fleming picked up his Nobel Prize and said, be really careful about how you use these medicines because the bacteria can become resistant, right? We just have never taken infectious diseases that seriously. You know, there's been plenty of pronouncements around the end of infectious diseases and, you know, and it's like, it's bullshit. These organisms are all around us. We've eradicated one, and the only one we've eradicated was because humans were its only, you know, its only source. So, yeah, I kind of disagree with you and agree with you at the same time. I think that's what I do. Anyway, thank you very much, everybody, for coming and for your questions.
And I guess we're only really touching the surface of this topic, so if anyone wants to carry on the conversation, Siouxsie and I will be upstairs in the cafe afterwards. I really want to thank you for coming down from Auckland to speak at this event. And there are really three big things that I really loved about your talk. Firstly, that you're doing wonderful, really cool, basic science to find new antibiotics. And secondly, the thing I think that we can all relate to is having the big picture about AMR and the more systematic approach, which obviously public health people love. And the third thing is about just being such a wonderful science communicator and really engaging the public and taking us all along with you. So thank you very much.
Thank you.